Hello, class. This is chapter 2, where we discuss the chemical level of life. All right, so to first have this discussion, we have to understand what matter is. Matter is anything that occupies space. So it could be you, it could be the computer or phone you're watching this video on, it could be the desk, it could it could be anything that occupies space exists. Matter exists in solid, liquid, or gas. So any solid, liquid, or gas is considered matter. And chemistry, very simply, is the study of how that matter study of that matter and how it interacts. All right. So the smallest unit of life we talked about in chapter 1 is the cell. Cell is the smallest unit of life, but the smallest unit of matter, which is even smaller than that, because uh, is the atom. The atom is the smallest unit of matter. So just like the cell is the smallest unit of life, it has all the properties of that particular form of life. We all human beings start off as a single-celled organism. The sperm fertilizes the egg in your mother's womb, and you become uh, that's conception, and then that cell just multiplies uh, a whole bunch of times, and eventually you, you become what we know as a human being. We all start off as a single cell. If you break down matter, the smallest unit of matter is called the atom. The atom, which is made up of protons, neutrons, and electrons. Right? These are subatomic particles that make up the atom, and so every type of matter, one of the simplest ones we've already mentioned is water, H2O. So an atom of water is two hydrogens, two oxygens, sort of bound together. It makes up a molecule, but each one of those, that's the smallest unit that makes up water. So if we look at the atom, it's broken down into protons, which are positively charged, neutrons, which have no charge, and then also electrons, which have a negative charge. And so atoms are generally neutral. You have the same amount of positive charge as you do negative charge, and these are floating around, and they make up what is called an atom. The electrons in an atom, and this is where we get into chemistry, and it can get a little bit intense, but the electrons are floating around the atom, whereas the neutrons and the protons are bound in the nucleus or together in the nucleus. The electrons that are floating around are organized. Remember, life is organized, and they're organized in different shells. And so the first shell, closest to the nucleus of the atom, generally has 2 electrons in it. And then the next shell has 8 electrons in it. And then you have a third shell, which can have up to 18 electrons in it. Atomic number. Those of you who are familiar with the periodic table, how we organize different types of elements, different types of substances. The atomic number is the number of protons. Remember, the number of protons balances out the number of electrons, the number of protons in an atomic nucleus. And so we can define a specific element: oxygen, carbon, hydrogen, sulfur, nitrogen. We define those elements by the number of protons in their nucleus. If they have a certain number of protons, it means they generally have that same number of electrons and that same number of neutrons. And for oxygen, it is a specific number of protons, neutrons, and electrons. And so that's how we define the element known as oxygen, by its atomic number, which is technically the number of protons. An element cannot be broken down to a simpler substance uh, by chemical means. And so we group those together in what is known as the periodic table, which has uh, elements listed by increasing atomic numbers. This is the periodic table here. We will talk about some of these elements over the course of this course. You do not need to know all of the elements in this periodic table, but there are some major elements, as you see here in the orange, the hydrogen, carbon, and nitrogen and oxygen. Those are major because they are very common in the earth and also in the body. And so you want to definitely pay attention to those. And then also the mineral elements, sodium, magnesium, potassium, calcium, phosphorus, sulfur, chlorine, are also very common in the body and things that we definitely will need to know and you will see again and again. So the human body is made up of the four major elements that we talked about: hydrogen, oxygen, carbon, and nitrogen, and then 7 mineral elements and 13 trace elements. There are also uh, this terminology known as an isotope. Isotope means very simply, you can have an atom with the same atomic number. So remember, atomic number is the number of protons in a nucleus. So the same atomic number, but different neutrons. So still have the same number of protons and electrons because they balance each other out generally, but you can have different numbers of neutrons. And that would be called an isotope. So hydrogen, for instance, has one, and then deuterium has two, and then tritium has three. So different numbers of neutrons, but the same number of protons and electrons. Radioisotopes are actually unstable isotopes, and that those extra neutrons uh, won't stay in that form for long, and they will release, which is called radioactive decay, and the isotope will assume a more stable form. One of the ways we can use that technology is, again, in tracers, and also kind of we can use it in imaging, but we can also use it in radiation therapy. Radiation can damage the structure of cancer cells, and so we can use that technology, that chemistry, to be able to attack certain parts of the body or to trace certain parts of the body. All right, so matter can combine, right? Different types of matter can come together. And so it can physically combine, but it can also chemically combine, right? So when we talk about physical combination, we're talking about one thing, still sort of maintaining its properties, but joining with another type of matter. But we also can have chemical combinations where we actually change properties. Think of oxygen, think of hydrogen, then think of water, which is very different from oxygen or hydrogen, right? So when hydrogen and oxygen come together and make H2O, make water, that's a chemical reaction that happens. But if you think of water and salt, for instance, you mix salt in water, you still have water and you still have salt. Even though it's mixed in, you can still taste the salt. It's still retaining its properties. And the water is still obviously the water. So you have salt water. Those things those are different ways that different types of matter can come together. And so we have three basic types of mixtures, right? So mixtures are retaining the properties of the matter. We have suspensions, we have colloids, we have solutions. A suspension, as you can see here, is, and the wonderful example is blood. If you've ever given blood, you've noticed when if you donate blood, for instance, and they get a whole kind of blood from your body, one of the things that happens is it's constantly being shaken, or as soon as you finish, they'll shake it up because blood is a suspension. You have different things suspended in the blood. You have red blood cells, you have white blood cells, you have platelets, you have different proteins that are suspended in the blood, not dissolved, suspended in the blood. And so if you were to set it to the side for a certain amount of time, some of the particles, some of the cells, platelets would drift to the bottom. Suspension. Colloids are evenly distributed and they will not settle out. Think of milk. So here you have, it's still a mixture, but it's evenly distributed and it won't settle out. The protein and the water in the milk stays mixed together. And then you have solutions, and this is where two or more components are extremely small, evenly distributed, and also will not settle out. So this one you could think of glucose and water, or you could think of salt water here, which stays mixed up, extremely small, and does not settle out. So those were physical mixtures, but you also, like I said, have chemical mixtures. All right, this is where you can have changes, a change in the chemistry. So here we have, um, so chemical bond is not a physical structure. It's an energy relationship or attractive force between atoms. You can have a molecule formed by chemical bonding between two or more atoms of the same element. And then you can have a compound when two or more atoms of different elements combine by chemical bonding. So two hydrogens bond together, make H2. That is a molecule of hydrogen. Um, but if you make, for an example, if you make this methane, which is carbon with four hydrogens bound to it, methane is a gas. Carbon is a solid, hydrogen is a solid, but methane is a gas. Oxygen that floats around in the air is generally in the form of O2, two oxygens bound together to make up an oxygen molecule. And the same thing with nitrogen. Chemical bonds follow what is called the valence electron octet rule. So atoms are generally most stable when their valence shell, their outer shell of electrons, has 8 electrons associated with it. And so here we have carbon dioxide. Everybody breathes that out, carbon dioxide. And that is, you have the 6 electrons, as you see here, in the outer shell of the carbon, of the oxygen. And then you have, and then it'll share 2 of the electrons from the outer shell of the carbon. So you have a covalent bond. And so basically the oxygen gets now access to 8 valence electrons and on each side because it's sharing 2 of the carbon here and 2 of the carbon here. Mm-hmm. Ionic bonds, which are different from covalent bonds we talk about sharing. So we'll share valence electrons so that we're both satisfied and both have that reach that magic number of 8. But in ionic bonds, one pulls the other. And so I think it's best seen in a nice picture here. Sodium or salt or sodium chloride is what some people scientifically we call it. Sodium has, as you see here, has 1 electron in its outer shell. Chlorine has 7, and so it's going to pull that 1 over to the chlorine and therefore fill its outer octane outer valence shell with 8, and then the sodium will just now have 8 in its outer shell because you removed the that one that was in its outermost shell. But now it has one more electron than proton. So what does that mean? That means that it's negatively charged. It has more as 10 electrons, and I'm sorry, it's positively charged. It has 10 electrons and 11 protons. And then chlorine on the opposite end, because it's pulled that extra electron away from the sodium, now it has one electron higher than proton. So it has a negative charge. And so you have this negative charged chlorine, this positive charged, and this positive charged sodium, and they bound together. And that's an ionic bond. So covalent bonds, so ionic bonds uh, pulling an electron, and then the two have opposite charges and they bind together. Covalent bonds are the strongest bond, and they share, as we talked about earlier, they actually share those outer electrons, right? So that gives, so each side is satisfied. The electron is sort of swirling around both of the atoms. And so they share these electrons to fulfill their outer shells. So they both get 8 in their outer shell by sharing the electrons. That's a covalent bond. Protons can attract electrons, known as electronegativity. And so depending on where, you can see we have it organized in the periodic table depending on where it is. The element's electronegativity increases from the bottom left to the upper right of the periodic table, fluorine being the most electronegative. So that's the ability to be able to pull electrons away from something else. You can also have non-polar covalent bonds. These are where you have the covalent bonding is equal, and so things are shared, but uh, you can also have polar covalent bonds. So non-polar, everything is shared. The electrons in the outer shell is shared. Outer shells are shared evenly, but you can have polar covalent bonds, which is a little bit different because now, even though it's a covalent bond and the electrons in the outer shell are shared, they spend a little bit more time towards one side than the other. That's considered a polar covalent bond. We see this with H2O. And so oxygen, because it has such a high electronegativity, can, even though it's a covalent bond, the electron will spend more time around the oxygen than it will around the hydrogen, therefore creating a slightly negative charge around the oxygen versus the hydrogen, which will have a slightly positive charge. But that gives water some pretty cool properties because of this polar covalent bond and this slightly negative oxygen and these slightly positive hydrogens. You get an attraction between that positive and negative, and it causes what we call surface tension in water because it's called they call it a hydrogen bond, and it causes water to have a surface tension. And so you can actually see that in the water molecule when you place a drop on a flat surface. You can see how it curves a little bit. That's due to those hydrogen bonds, which are due to the fact that there is a polar covalent bond, and that the oxygens are slightly negative and the hydrogens are slightly positive, and there's an extra bond in there besides just the covalent bond. There are different types of chemical reactions that can happen. Chemical reactions when chemical bond is formed or broken or rearranged. And so we have different chemical reactions that happen. Remember, metabolism is the summation of all the chemical reactions happening in your body. And so generally we have reactants on the left side of the equation and products on the right side of the equation. So we have hydrogen plus oxygen equals H2O, water, for instance. The hydrogen plus the oxygen, that would be the reactants. The water, that would be the product of the reaction. Right? Some reactions are reversible, they can go in either direction, and some are irreversible. Whenever we talk about chemical reactions, we need to talk about energy as well. Energy is the capacity to do work or to fuel chemical reactions. And so there's oxygen floating around in the air right now. There's hydrogen floating around in the air right now. But why isn't it just coming together and exploding and drops of water just coming from nowhere? Well, it requires energy for that to happen. And so potential energy is energy that is stored and can be used for work at some point, released and used for work. And then you have kinetic energy, which is potential energy that has been released or is doing some type of work. Right? We also have chemical energy, the energy inside of bonds. We have electrical energy. We have mechanical energy, energy directly transferred from one object to another. When you push something and it starts rolling. There's also different types of reactions. There's endergonic reactions. That means reactions that require the input of energy for those reactions to happen. And then you have exergonic reactions, reactions that release energy, right? So if it takes energy for that reaction to happen, it's endergonic. If it releases energy, it's exergonic, right? And that's going to be it's going to be very important. There's also uh, different types of reactions, catabolic reactions, reactions that break down something. If I was taking a huge carbohydrate and breaking it down into glucose, that's a catabolic reaction. There's exchange reactions where I could be transferring things like electrons from one um, one compound, one molecule to another. That's an exchange reaction. There are anabolic reactions, reactions where I'm building things up. You build new skin after your body suffers an injury. When you cut yourself and then your body needs to build new skin, that's an anabolic reaction that needs to happen. Here's an example of a catabolic reaction. Just AB is your reactant, and then A plus B is your product. And so something is broken down, and this is usually exergonic. So usually releases energy when you break those bonds. Oxidation reduction, these are exchange reactions. So something that is oxidized means that it loses electrons. Something that is reduced means that it gains electrons. Now you may say, well, if we call it reduced, but it gains electrons, that doesn't make any sense. But think about it. Electron has a negative charge. So if I'm transferring electrons from one compound to another compound, if I'm transferring electrons, it's actually reducing the charge, right? So these are these are reactions that generally release a lot of energy. And then you have anabolic reactions that we talked about, building things up, anabolic reactions. A plus B yields AB, for instance. And these reactions generally require energy. When you build, you require energy. Think about if you go lift weights and then your body wants to build some muscle. Uh, you gotta feed it protein. You gotta feed it carbohydrates. Your body needs energy to be able to do that. So where do we get that energy from? Well, there's from the food we eat is the simple answer, but also your body has ways to reduce the amount of energy needed for a reaction to happen. Right. So the energy that is required for a reaction is called activation energy. Right. That's the energy that we need to get to be able to have this reaction to happen. This is a diagram that just talks about it. So you have your reactant. So think about the the hydrogen floating around in the air, the oxygen floating around in the air. It would take a significant amount of energy. Heat is a form of energy. Electricity is a form of energy. A significant amount of energy for that hydrogen and that oxygen to come together and to eventually make what is called water, right? So to get to so to get to that point, that transition state would require a lot of energy. One thing that we can do is change some of the factors and reduce the amount of energy needed to be able to have that reaction happen, right? So if we increase the concentration, put more hydrogen and oxygen closer together, if we increase the temperature, heat, that's energy, um, we can change some of the reactant properties or even the presence or absence of a catalyst. Now, what is that? Well, we're about to see. Right, so concentration increases more times the reactants are interacting with each other. Temperature also increases how much those reactants are moving. Catalyst is going to be very important in anatomy and physiology. It increases reaction rate by lowering the activation energy. But a catalyst is not consumed or altered in the reaction. In the human body, since this is anatomy and physiology, in the human body, we have biological catalysts, things that help our body be able to reduce the activation energy so that our body's reactions can happen more likely. These are called enzymes, and they are proteins that speed up reactions. They're very specific, but they don't alter the reactants or products, and they're not permanently altered in the reaction. Right? So with a catalyst, we can, in the body, it's called an enzyme. That's the catalyst in our bodies. But with the catalyst, we reduce the activation energy, and therefore reactions, the certain reactions are more likely to happen, and therefore your body can get the products that it wants. This is an example of an enzyme, a protein that is an enzyme in the body. It will bind to reactants and allow them to be able to come together and so that they can have a reaction. And then the product is released. There are some deficiencies, and you'll see in this class, we'll talk oftentimes about different deficiencies. But if you lack a certain enzyme in your body, then your body cannot then it cannot do certain types of reactions. Then your body will have trouble in that particular area. Tay-Sachs disease, for instance, deficiency of hexosaminidase and gangliosides. You learn about ganglions in anatomy and physiology too. Gangliosides accumulate around neurons of the brain, and fortunately usually leads to death, but they have a deficiency in it, in this particular enzyme, the hexosaminidase. So your body, like we said, metabolism are all the chemical reactions that happen in your body to keep you alive. Your body has to do thousands of reactions for you to stay alive. And when we don't have an enzyme, we don't have the reactants, or we're not making the certain products in our bodies, it can become very problematic very quickly. All right, so let's switch subjects a bit, go into inorganic compounds. So biochemistry, the chemistry of life, we need to study inorganic and organic compounds. It sounds complex, but it's not really complex. Inorganic compounds, very simply, do not contain carbon bound to hydrogen. So if you think of the term organic in the grocery store, it tends to mean they didn't use pesticides when they grew this uh, grew this watermelon, grew this chicken. In anatomy and physiology, or in the science world, when we say organic, it means that you have somewhere in that equation carbon bound to hydrogen, right? So things like methane are organic. Things like glucose is organic. But things like water, H2O, are not organic because water does not have carbon bound to hydrogen. Let's talk about water. Water is inorganic. That means it's not organic. Makes up 60 to 80% of the body. It's Water is very necessary for life. Has a high heat capacity, able to absorb a lot of heat without significantly changing temperature. It can also evaporate and remove heat from the body, cushions the body, acts as a lubricant. It's also the body's primary solvent. You know, a lot of things dissolve in water, right? Things that are hydrophilic dissolve in water. So, we all know salt dissolves in water, and a lot of ionic and polar covalent solutes. So ionic, NaCl, ionic, polar covalent. That means it's a covalent bond, but uh, one side it has a much higher electronegativity and pulls the electrons away more from the other side. So things that are polar covalent, or things that are ionic, dissolve very well in water. Hydrophobic solutes, things that have covalent bonds, don't do as well in water, don't dissolve in water. A lot of your fats do not dissolve in water. We call those hydrophobic solutes. So fats, oils, they don't dissolve well in water. Acids and bases. So when we study acids and bases, we're studying um, hydrogen ions. So water molecules in solution will dissociate and break apart and form hydrogen ions and positively charged hydrogen ions and negatively charged hydroxide ions. And so we study the positively charged hydrogen ions and study that, and that gives us uh, an idea with respect to how acidic or how basic a solution is. So an acid is something that hydrogen ion or a proton donor, and a base is a hydrogen ion acceptor. Right? So if I put in an acid, it generally donates a hydrogen ion. Hydrochloric acid, HCl, it will donate a hydrogen. And, and if I put in a base, it'll accept a hydrogen. And this is what we call a pH scale, right? Simple way of representing hydrogen ion concentration, right? So if something donates a lot of hydrogens, it's very acidic. And so it will have a very low number on the pH scale, as you see at the bottom of the red one. If something can receive a lot of hydrogen, it is very basic and it will have a high number on the pH scale, as you see extremely basic on the blue side, a high pH. And you can see things that are very basic and things that are very acidic and in between. Notice that blood is very close to water and milk, sort of very close to the middle. So very almost neutral, not neutral, but pretty close to neutral. A buffer, which we will talk about later in more detail, but just want to introduce it here. A buffer is a chemical system that resists changes in pH. And so the idea for a buffer is, even though I'm taking in something that is acidic, like you can drink lemonade, for instance, which is acidic, your body's pH, your body's pH will not change that much. Your blood pH won't change that much. Um, your body, your blood pH has to stay between 7.35 and 7.45. You may think, wow, that's a small range. So it has buffers that are in place that allow you to be able to take in something acidic or something basic, and the overall pH of your body doesn't change too much. That's what buffers are very much known for. And so we have this carbonic acid bicarbonate buffer system, which is a reversible reaction, can go in either direction. So if I have too much acid, I can make more carbonic. I can make more bicarbonate ion. If I have too much base, I can make more carbonic acid. I can balance it out. There's also the term, you're going to see the terms salt and electrolytes. Salts are any metal, cation, non-metal anion held together by ionic bonds. We'll see those. We'll see electrolytes, which can dissolve in water to form cations, anions called electrolytes. And they are electrolytes because they are capable of conducting an electrical current. And then there's also organic compounds. So all that was inorganic: water, salts, electrolytes, acids, bases, inorganic compounds. But we also have quite a few organic compounds that are very important. Like we said, organic compounds have carbon bound to hydrogen in their formula. And the different types of organic compounds that we have are in our bodies are carbohydrates, lipids, proteins, nucleic acids. They generally are built by what is called a monomer. So a single subunit which combines to make larger polymers. And that'll make sense as we kind of go through it. Hydrolysis is a catabolic reaction, uses water to break polymers into smaller subunits. So the opposite of that is dehydration synthesis, which links monomers together using water. Right? Remember, we said that anabolic reactions, reactions that build, generally require energy, a lot of energy. All right, so different types of organic compounds. Organic meaning it's somewhere in the formula there is carbon bound to hydrogen. So carbohydrates, everybody's probably heard of carbohydrates, low carb diets, all those kinds of things. You need carbs for energy, blah, 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 blah. Carbohydrates have carbon, hydrogen, oxygen, and they generally provide energy to the body. Glucose, fructose, galactose, ribose, all in the body. Those are monosaccharides, monomers from which all carbohydrates are made. So monomers combine together and make polymers. So the monosaccharides are the monomers of the carbohydrates, and the monosaccharides will combine together to make larger polysaccharides. Right, so these are the monosaccharides. Here, glucose is probably the most common that you know about glucose. So these are monosaccharides, sort of the building blocks of your bigger polysaccharides. So you can combine two monosaccharides and make a disaccharide using what is called dehydration synthesis. You lose a water molecule in the process. It's going to require energy for this to happen. And then you can also make polysaccharides. So, for instance, glycogen is a bunch of glucose monosaccharides bound together, and you can make what is called glycogen, which is stored in your skeletal muscles and your liver cells. That's how energy is stored in your body. This is your polysaccharide, glycogen. See all those glucose molecules bound together. We also have lipids, another organic compound that is in the body. It's nonpolar hydrophobic molecule, carbon and hydrogen. Fatty acids are lipid monomers, 4 to 20 carbon atoms. Can have double one one or more double bonds. This is your saturated fatty acid, what it looks like chemically. Saturated meaning there's no double bonds in the carbon hydrogen chain. Monosaturated, you can have one double bond. These are generally liquids at room temperature. Polyunsaturated fatty acids, you have multiple double bonds. So it's unsaturated, means it doesn't have the maximum number of carbons, maximum number of hydrogens bound to the carbon, because some of the carbons are double bonded. Different types of fatty acids here. You don't really need to remember this, but this is different types of fatty acids. Omega-3s, saturated fats, trans fats. Some good to know information for your personal health. Lipids are 3 fatty acids linked by dehydration synthesis to a modified carbohydrate. And that's where we get the term triglyceride. Tri, 3 fatty acid chains, that's the tri, and glyceride bound to a glycerol molecule. Phospholipids, which you're going to see in the next chapter, have a glycerol backbone and 2 fatty acid tails. We see this structure in the cell membrane. That's the structure of the phospholipid. You also have steroids and cholesterol, which are also forms of different forms of lipids. Proteins, also organic compounds found all throughout the body. You have fibrous proteins, which are rope-like strands, and you have globular proteins, which are spherical. Proteins are primarily made up of amino acids, which are bound together. Then they begin to fold on each other and are held together by hydrogen bonds. They fold again, twist and fold, and stabilized again by hydrogen bonding. And then they can even, one polypeptide chain can link together with another polypeptide chain and form a larger structure. And that's what makes up a protein. Proteins, one problem with proteins is that they can denature based on the heat or the pH. This is why your body needs to stay at a specific temperature. And your body needs to stay at a specific, what's the word? pH, specific pH, yes. Can't get too acidic or too basic. You can't get too hot or too cold. On the inside. On the outside, yes, but on the inside, your body maintains temperature because of protein denature, the ability of protein to denature. Protein, those amino acids make up a chain, and then it folds on itself, and it folds and it bonds and it folds with other chains and makes up a structure. Well, that folding is very important into the function of that protein. And so things like heat, things like pH, things like chemicals, things like drugs cause that protein to lose its shape, and then it loses its function. Remember, in anatomy and physiology, anatomy is form, physiology is function. Form and function are very much intertwined. If we change the form, we're going to change the function or disrupt the function. And we also have nucleotides, another form of organic compound. Nucleotides are made up of nucleic acids, and they uh, are the structure that's very important in our DNA and RNA. They generally are a nitrogenous base, some 5-carbon sugar, and a phosphate group. Types of nitrogenous bases are adenine, guanine, which are the purines, and you have cytosine, uracil, and thymine, which are the pyrimidines. These are the bases that help make up the nucleic acids. One of the most famous ones is adenosine triphosphate. We're going to learn about this in this class. Adenine attached to ribose and 3 phosphate groups. And this is one of the main sources of chemical energy in your body, has a lot of potential to do work. ATP, adenosine triphosphate, is highly endergonic reaction due to negative charges on phosphate groups. To form the ATP, it's endergonic, so it requires energy to form it, but then it can release a great deal of energy when you break the bond. Right? So it takes energy, so your body can take energy from your food that you eat and form ATP to store energy. But then when your body needs energy, it can use that ATP, break that bond, and release that energy to be able to do different types of reactions that are necessary in your body. Notice that second line there says production of large quantities of ATP requires oxygen. This is the reason we breathe air. So we need energy from the food we eat, but we also need oxygen to be able to make ATP, which you'll learn about. You'll learn that process later in anatomy and physiology. DNA, which DNA is the genetic, contains the genetic material. It's made up of nucleotides and nucleic acid. So we have deoxyribose, which is the backbone of the strand of DNA. You have adenine, guanine, cytosine, and thymine. Forms this double helix structure held together by hydrogen bonds. We'll learn more about DNA later on. And then we also have RNA, which is a single strand of nucleotides, which also contains genetic information. When we try to copy DNA, we copy it into a single strand of RNA, but it has a little bit different nucleotides as uracil instead of thymine. So a little bit different structure. Transcription is RNA copies. So we take a section of DNA and copy it. It's called transcribing that DNA. And then translation is we use that use that RNA that we have. So DNA is super long. We may only need just a short section to code for a specific protein. So we copy this section, make a copy. The copy is called RNA. That's transcription, getting the RNA copy. But then we take that strand of RNA and we translate it into the protein. So use that to use that as the code to make the protein, and that's translation. And that's how we go from DNA, genetic material, to protein, which is our body needs. We'll explain that in more detail. Thank you very much.